let me first start by thanking Elliot for sending that letter to Graham, and Graham for reading the letter, and also for everybody involved in this, people at LBNL, Larry Conrad, everybody else, David Greenbaum, Patrick Smith, everybody, for all the hard work on this. Because I'm going to predict that the community on campus that this is going to impact the most and be most transformative for is going to be the biologists. And why is that? That's because I think people in astrophysics, they've been struggling with these problems and have been working with high-performance computing for a long time. But biology is these days being completely transformed into becoming much more computational. And many people in biology, the biological community, has not really geared up for that, has not been prepared for that. So why is it that we're becoming so computational now? Well, it's mostly driven by the new availability of data. So this is a figure that if you're a biologist, you probably have seen many times. What it shows is Moore's law here. And then it shows the price of sequencing, of DNA sequencing in comparison to Moore's law. And what you can see is what happened in around 2008 or so is that the price of DNA sequence dramatically dropped. Okay, so we can sequence genomes now much, much more cheaply than we could previously. So when I was in graduate school, we always asked these questions by our professors, you know, how would you design an experiment to do this and that? And we were saying, well, how much money do I have to do it? And they say, well, don't worry about the money. And we would say, well, let's sequence the genomes then and solve the question. And say, no, that's cheating. You can't do that. Okay, so today we can just do that. Today, all the graduate students, they can cheat. They can just sequence the genomes and answer many of these questions. And it's really changing how we do biology. So let me start by giving you a few examples of the kind of things that we're doing, how biology is being changed. So one big area that's completely changing is cancer research. So what people are doing now is that they're taking cancer tumors and they're deeply sequencing the DNA in these cancer tumors. And by doing that, they're able to identify exactly what the specifics of that tumor, so that when you do diagnostics of a patient, you can figure out exactly which kind of cancer that is. And then you can individualize the treatment much better. And then, in fact, cancer cells that are circulating in the blood. So in the future, what we think we'll be able to do is to just take a sample of blood from an individual that we think may have cancer, diagnose whether the individual has cancer, and then make an individualized treatment of that cancer just based on the blood. But to be able to do that, of course, we need to be able to analyze massive amount of data sets, massive amount of data of DNA sequencing data. A lot of the research that has been going on over the past few years in genomics has also focused on finding the genetic variants that are responsible for heritable diseases. And one disease in particular, type 2 diabetes, literally hundreds of millions of dollars has been spent on trying to find what's the genetic underpinnings of that disease. Why are we interested in that disease? Well, because it's a very, very common disease. It's a huge burden on society because it's so expensive to manage, but also because it just impacts so many people's lives. So we really want to understand the genetics. If we can understand the genetics of this disease, we can devise better treatments. And so to do that, what are we doing? Well, we are sequencing the genomes of thousands, hundreds of thousands of people. And then we're trying to figure out what are the genetic factors that determine who gets type 2 diabetes and who does not. And now that you're all sitting here and you can't run out, I'm going to give a little example from my own research. Squeeze that in here. So this is something, a paper we have in press in Nature that's going to come out next month, where what you see on the x-axis here, that's the different positions in the genome. And on the y-axis, what you have there is a measure of statistical confidence in a particular genetic variant being associated with diabetes. This is for a group of individuals from Greenland. And what you can see, there's one peak in the genome. And if you have a particular genetic variant there, the chance of getting type 2 diabetes is more than tenfold increased. So in Greenland, we can now predict pretty much based on the genetics, if you're at the age of above 60, who has type 2 diabetes and who does not have type 2 diabetes. Of course, there are only 60,000 people that are Inuits from Greenland. So it's not so many people's lives we will affect by this. But it's an example of how we can find the genetic underpinnings of some of these diseases. And by doing that, we can maybe also then develop better drugs and be able to treat these diseases in a better way. So another area where people are using a lot of DNA sequencing and where that really has changed is also in looking at various evolutionary problems and, for example, human history. So another little project I'll entertain you about is this sequencing of the remains of an individual, a boy that died 12,500 years ago in North America, a Native American. And what we did there was we sequenced the genome of this individual. And by doing that, we could show that this individual belonged to a population that is ancestral to all modern Native Americans. And in fact, from that, we are able to piece together the history of how the Americas were populated, people moving into the Americas about 15,000 years ago, and that the big culture that established shortly thereafter was known as the Clovis culture, that culture is really ancestral to all modern Native Americans. So DNA sequencing is not just being used for understanding molecular biology, for understanding diseases, but for many, many, many different things, environmental sequencing, understanding what's out in the environment. We're doing, for example, we're looking at ice cores in Greenland and figuring out what was the environment 400,000 years ago in Greenland, because we can get DNA out of the ice cores. So projects like that are changing biology by doing all this deep sequencing. Okay, but the cost of this is increased computational demands. So now any biologist can now solve some of the problem by doing a little experiment and then generate some terabytes of data. But they're not been educated to deal with the data, and they don't have access to the computing to deal with this. So the demands for computing is really twofold for the biologists. There are people like me that develop methods. On a daily basis, we may be like the astrophysicists. On a daily basis, we need access to high-performance computing to test out our methods, do preliminary analysis, and so on. But then there's a much larger community of biologists that need access to high-performance computing maybe a couple of times a year. Maybe just do something that will take them analysis that will take them 2 days to do on a cluster, but they could never do on a desktop computer. And they're pretty much at a loss now. Often they go to other universities, establish collaborations with people that sit at other universities. And that's, I think, really where the BRC really will make a huge difference for the biology community with the condo model for computation, because it fills that need. People like me, we can buy notes now, we can get help with administration, exactly like the same way they do in astrophysics. And then all the other biologists that occasionally have a need for some high-performance computing, they can get some of the remaining cycles that are available for them. So I predict that the BRC will really fundamentally transform how many people do science on campus. It also helps us in one last way. We might be able to actually get funding for some of the computing, because that's been a problem in some of the previous grant proposals for high-performance computing clusters at Berkeley. We always being told that, well, actually, we would like to give it, the money to you, but you have no institutional support. How are you going to maintain this cluster? This is inviable. We're not going to give you the funding. Now we have an answer to that. So I think we have very high hope that we'll actually be able to bring in more computing resources to UC Berkeley through this. And in fact, we already worked together with David Greenbaum and other people, with the people at LBNL, on a proposal that's being processed right now at the NIH for bringing a cluster to Berkeley. So let me end again by thanking the people involved in this, Elliot for his letter, and everybody else. Thanks.